So let's get going. So we're focusing on the integumentary system. And when we think of the integumentary system, it's more than just skin. Yes, skin is a major component of the integumentary system, but there's also hair, there's also the nails, fingernails, toenails. There's also going to be the glandular secretions associated with the integumentary system as well. And as we look at all of these components of the system, it's surprisingly complex. Because our skin is on the surface of our body, it's exposed to lots of damage, so we're going to need lots of repair mechanisms in place throughout our adult lifespans to maintain our skin. And for those of you who study skin, that's known as the field of dermatology. The root word there, derm, referring to the living layer of your skin, ology meaning the study of. So here, it's just a basic diagram showing an idealized cross-section of thin skin, thin, hairy skin. And we'll talk in more detail about all these structures that are associated with the skin. So let's focus on our skin and subcutaneous tissue. As we're looking at our skin, it could be considered an organ. What's the definition of an organ? If we think back to that Chapter 1 presentation, an organ has two or more tissue types working together for a common purpose. And within our skin, we have multiple tissue types. We have the stratified squamous of the epidermis. We have the dense irregular connective of the dermis. And then we have some simple cuboidal and stratified cuboidals in some of the glandular secretions or glands of the dermis. So it is, by definition, going to be considered an organ. But this is one of those like semantic or pedantic things. Depending on who you ask, the skin is the largest organ or the liver is the largest organ. Sometimes we also think of the skin as more of a membrane because it's a barrier that keeps things out of our body and keeps things in our body. But it meets the technical definition of an organ. It's worth mentioning that within the third layer, that hypodermis, the layer that's deep to the dermis, there's two primary tissue types, notably adipose tissue and areolar tissue. So what's our skin do for us? It helps us resist trauma. That's a nice way of saying that it's just a physical barrier for us. Our epidermis is keratinized, so those squamous cells on the surface of our body are filled with a tough, waterproof protein, hence the name keratin, or the word keratin. Those cells are filled with the keratin protein. And it's more than just the keratin protein. We also have other antimicrobial components of our skin. We have what's known as dermacidin and defensin. As the name implies with defensin, it's a defensive protein. These proteins serve to selectively kill off microorganisms on our skin. We also have something known as the acid mantle of our skin, or you can think of it as more of a patina or a layer of acidified tissue. Our skin is acidic. We have uric acid constantly being secreted onto our skin from our sweat. One of the things that makes our sweat stinky. And that uric acid acidifies our skin and makes it inhospitable to many of the microorganisms that would like to get into our body where we have a more neutral pH. Part of the acid mantle is the high saline content. Your sweat is salty, and when your sweat gets on the surface of your skin, the water evaporates into the air, and that salt is left over. So you have highly salty, highly acidic skin that makes it difficult for most microorganisms to live on your skin. The keratin component of our skin serves a very important purpose. It helps to waterproof the epidermis. We don't live in ponds. We are not aquatic organisms. So because we're terrestrial, we live on land, we need some way to keep the water in our bodies. And that keratin within the epidermis is a key function of that. We also have highly developed kidneys that help us not urinate out our water. Within our skin, we have a pigment, two pigments primarily, that we make to help absorb ultraviolet radiation. If ultraviolet radiation gets to the nucleus of a cell, there's a chance that it can form something called the thymine dimer. If there's two thymines, or two T's side by side in the DNA, and those two thymines are hit by a photon of UV radiation, the thymines are going to have a new covalent bond formed between them, and now we just had a mutation introduced into the genome. And when we think of our skin, it also helps us with vitamin D synthesis. This time of year, most of us in here are probably suffering from a shortage of vitamin D because we've been bundled up. We haven't had a lot of direct sun exposure for the last several months. And our skin is a primary organ to help us with the process of synthesizing vitamin D. Now, we don't only make vitamin D in our skin. That's a common misconception. Vitamin D is partially processed in our skin, but other parts of our body that helps to process the vitamin D include the liver and the kidneys. So the skin is just one of the places where we process that vitamin D. We also use our skin for sensation. We have a lot of sensory organs. We have thermoreceptors. We have pressure receptors. We have pain receptors. We have all of these sensory receptors on our skin to help us have information from the external environment to be sent to our brain to be processed. Within our skin, to help us with thermoregulation, we can have vasoconstriction and vasodilation. When we think of vasoconstriction, that's going to be when we take the blood vessels and constrict them or make them smaller. Vasoconstriction is when we reroute blood from the superficial tissues of our skin and send that blood to the core organs of our trunk and our head to maintain core body temperature. And then we can also have vasodilation. And in those situations where we have vasodilation, we dilate or open up the superficial blood capillaries so that more blood is redirected to the surface of our body so that we can radiate excess heat from our core organs to the outside environment. This is just one way that we can help to maintain homeostasis. We also have perspiration. When water is on our skin and the water goes from a liquid to a gas when it evaporates, there's the heat of vaporization. There's an energy expenditure associated with that evaporating of the water. And as the water evaporates, it's going to absorb heat energy from our skin, and that heat energy will dissipate with the steam. And then we also have a lot of nonverbal communication. You know, you can look at someone's face, look at their skin, and have an idea of what they're feeling, what they're thinking. So that's another really important function of the skin. And it's not just facial expression, it's also the color of their skin. Are they flushed? Are they pale? Do they have kind of a greenish pigmentation to the skin? You know, does it look like they're going to throw up? I mean, there's also some color associated with that nonverbal communication. As we look at the most superficial layer of our skin, that most superficial layer is known as the epidermis. Epi meaning above. So when we look at that word epi, think above. What is this layer above? It's above the dermis. And the epidermis is going to be made of a stratified squamous epithelium. Now within our epidermis, most of the cells are not metabolically active. Most of the cells are going to be dead. But the deeper you go within the epidermis, the closer those cells get to the nutrients of the dermis, from that highly vascularized dermis. So that stratum basale, or the deepest layers of the epidermis, are going to have living cells associated with them. Our epidermis lacks blood vessels. It is an epithelial tissue, being a stratified squamous epithelium, so it's going to be avascular by definition. So we're not going to have a lot of metabolic activity in most of the layers of our epidermis. And then we're also going to have very few nerve endings in our epidermis. And that's a good thing. That means that, you know, you can scratch your skin and it doesn't hurt when you shed off a couple of layers or maybe a dozen layers of squamous cells from your epidermis. Within the epidermis, there are multiple cell types. We have a lot of stem cells. Now, last chapter, chapter 5, we talked about the different kinds of stem cells. We have the totipotent, multipotent, pluripotent, and induced pluripotent. Of those 4 stem cell types, the stem cells in our skin, we as adults will have multipotent stem cells or adult stem cells, which is a nice way of saying that these stem cells in our skin can turn into other skin cells, but they're not going to turn into a tooth or a muscle or other random tissue types of our body. So these stem cells of the skin will differentiate into other cells of the skin. And of the cell types that they form, the keratinocyte is the most common kind formed by the multipotent stem cells that are present in our epidermis. A keratinocyte makes up the vast majority of the epidermis. So when you see all of those flattened cells, all of those squamous cells within the epidermis, you're looking at mostly a bunch of keratinocytes. Another notable cell type in the skin is the melanocyte. And the melanocyte is typically going to be located within the stratum basale of the epidermis. And generally speaking, with few exceptions, we all have the same number of melanocytes, or the same density of melanocytes. So you can think of it as the same number of melanocytes per square centimeter, regardless of our skin color. And these melanocytes can be activated and deactivated, or upregulate and downregulate melanin production based on UV exposure. So if we expose the melanocytes to ultraviolet radiation, that upregulates melanin production, and then they start making more pigments, which are then going to absorb more ultraviolet radiation and minimize the amount of thymine dimers, or DNA mutations, that we build up over time. We're also going to have tactile and dendritic cells. When we think of our tactile cells, these tactile cells are going to be in the stratum basale because they're metabolically active and they need to have nutrient delivery and removal. And they're typically going to be a light touch receptor. So when we think of these tactile cells, think of very gentle or light touch. They are not going to be for deep pressure sensation. If you've ever had, maybe your foot's fallen asleep, and you try to walk on your foot after your foot's fallen asleep, you've lost innervation to the tactile cells of the epidermis, but you still have those deep pressure sensors that are getting enough blood supply to be active. And it feels really weird. It happened to me this weekend. You could take a step and you feel the pressure deep down in the middle of your foot, but you don't feel the skin or the epidermis of your foot at all. And that's what happens when those tactile cells are partially deactivated for a short period of time. And then finally, we have dendritic cells. Dendritic cells are a kind of white blood cell, a kind of macrophage that's present within the epidermis. The purpose of the dendritic cell is to phagocytize, to swallow pathogens that are trying to burrow through your epidermis to get into your body. If we look at the root name of the dendritic cells, it's dendrite, which refers to the branching of a tree. A dendrologist is somebody who studies trees. So these dendritic cells have lots of kind of extensions coming off their cell body that look like tree branches as they reach out in between the stratified squamous cells to phagocytize potential pathogens. So here are those cell types we were talking about. In green, we have the dendritic cell. We also are going to have a tactile cell in blue. In brown, we have the melanocyte. And then making up most of the cells of the epidermis on the screen, we're going to have those keratinocytes, the cells that make and fill themselves with keratin. Now, as we're looking at our skin, our epidermis, we can have thick or thin skin. All skin will have at least 4 layers to its epidermis. Thick skin will have a 5th layer to its epidermis. So, starting from deep and moving to superficial, let's talk about those layers of the epidermis. We have the stratum basale, which is the deepest layer, and it's 1 layer of cells thick. It's going to be directly against the cells of the dermis, and it's going to receive a lot of nutrients. Because the stratum basale receives lots of nutrients from the underlying dermis, it's going to have lots of metabolically active cells. This is where those multipotent or adult stem cells of the skin are primarily going to be located. And as those multipotent stem cells divide, we get more and more keratinocytes, and we'll have a few tactile cells and a few melanocytes present in this layer as well. The next deepest layer, so as we went from deepest, now we're moving our way superficial, is the stratum spinosum. Within the stratum spinosum, we're going to have those keratinocytes joined together by the extracellular junction of a desmosome. So the keratinocytes are going to be anchored together, but still have some flexibility. There's also going to be a lot of tight junctions. And those tight junctions, to review, are waterproof junctions. So in addition to the cell being filled with keratin and it being very difficult for water to move through the cell, water has a very hard time moving in between the cells because of those tight junctions. And why do we call the stratum spinosum the stratum spinosum? Well, under the microscope slide, some people feel that they look spiny. I personally am not one of those individuals, but I guess if you squint your eyes and look at it upside down, they look spiny to you. As we're moving superficially, so we're heading to the next most superficial layer, we have the stratum granulosum. The stratum granulosum is going to be closer to the surface. It's above the stratum spinosum, and it's going to typically be about 3 to 5 layers of keratinocytes. The key characteristic of the stratum granulosum is that the keratinocytes are now going to be filled with granules. And when you're looking at histology in lab, you probably noticed those granules, those dark purple speckles that appeared within the cells of the epidermis. Stratum lucidium is only present in thick skin, And this layer is very thin and pale. It's called the lucidium based off of the root word for light, or lucid. Because on a microscope slide, light tends to travel right through this layer of the stratum lucidium, and it's white in appearance, hence its namesake, or the lucidium for its namesake. And within this stratum lucidium, the keratinocytes are going to have an additional protein. In addition to keratin, there's also elidin. And this protein helps the keratinocytes to stick together for a longer period of time. And because these keratinocytes are extra adhesive to each other, or extra stuck together by this additional protein, we can have a really thick stratum corneum. Thin skin doesn't have the stratum lucidium. It lacks that clear protein, and consequently, the stratum corneum of thin skin isn't going to be as many layers thick. Now, when we look at the stratum corneum, it's really thick, depending on the tissue type we're looking at. With thin skin, our stratum corneum can be more on the 5 to 15 layers of keratinocytes thick. But for thick skin, it is very common to have 30 layers of squamous cells within the stratum corneum. When we look at the stratum corneum, it's going to be responsive. Some of you, you don't have to raise hands in here, but some of you were probably a thumb sucker when you were younger. And one of the key characteristics of thumb suckers is that they develop thick skin on the side of their thumb as their lower incisors are constantly rubbing against their thumb, and that thin skin can transform into thick skin because of repeated abrasion and repeated frictional forces. So in our body, we have the bottom of our feet and the palms of our hand, the two parts of our skin that are constantly exposed to frictional forces, because you're always walking on the bottom of your feet, and you're always grabbing things and rubbing the palms of your hands on them. And because of that constant abrasion, constant frictional force, we have the thick skin built up in those parts of our body, and consequently need lots, up to 30, layers of dead cells. It's worth mentioning that if you have the callus or a corn, you can have over 30 layers of dead cells on your stratum corneum. Or if you have maybe cracked heels, you'd have over 30 layers of cells. So, as we focus on the keratinocyte, the single most important cell type of our epidermis, it's always going to start out with those adult stem cells in the stratum basale. And that adult stem cell, that multipotent stem cell, will divide. It'll go through a mitotic phase. And there'll be 2 daughter cells. One daughter cell will remain within the stratum basale. The other daughter cell is going to start migrating upward. The one that migrates upward is going to be transformed into a keratinocyte. And then the daughter cell that remains within the stratum basale will remain as a multipotent stem cell, so that we can have new skin regenerated later on in life. And as these new keratinocytes are constantly pushing the cells up towards the surface, the older cells are going to move farther and farther towards the surface to make room for the new keratinocytes. In terms of what stimulates reproduction of these multipotent stem cells, it's typically going to be elbow room. They need the physical space to reproduce. So if we have some kind of an injury to our skin, all of a sudden now there's physical space to grow into. There's some new real estate that's been freed up, and we'll have an accelerated rate of division of these multipotent stem cells. But assuming you don't have any kind of damage to your skin, these multipotent stem cells are going to be physically crowded out, and the physical crowding down-regulates the rate of mitosis so that you don't have giant, like, warts or corns or calluses randomly growing throughout your body. Occasionally, there will be something that is going to upregulate the rate of mitosis, typically the human papillomavirus, but that is a homeostatic imbalance. That's an exception to the rule. As those keratinocytes move superficially, move upward, they tend to flatten, they tend to become anucleate, they lose their nucleus, and they're going to be filled with keratin. In terms of how long it takes to make a keratinocyte that makes its way to the surface, it's going to be about a month or a little bit over a month, 30 to 40 days. Generally speaking, when you're young and healthy, this happens super quickly. And then as you age, the rate of cellular division declines, you have fewer multipotent stem cells, and it takes longer to regenerate your skin. If you hadn't put it together yet, a callus or a corn is an extra thick layer of keratinocytes, usually fairly localized, and in response to repeated abrasion. Now, within the stratum granulosum, the keratinocytes are going to have some additional proteins released, one of which is filaggrin. And when we look at these filaggrin, the filaggrin protein is going to have a lot of filaments associated with it, hence the root word fil for filaments, and it's going to bind the keratin together. And within that stratum granulosum, the cell is also going to have a lot of proteins just beneath the cell membrane to help maintain the integrity of that cell membrane. And we'll have lots of lipids that intermingle with these proteins. They'll be directly beneath the surface of the cell membrane and directly above the surface of the cell membrane. Consequently, not only do we fill the cells with waterproofing proteins, we also are going to cover the cells with a hydrophobic chemical compound to further enhance the water-repelling abilities. Eventually, as the keratinocytes are moving superficially, all of the organelles, the nuclei, the mitochondria, everything is going to degenerate. There'll be self-induced autophagy, where those organelles are going to be consumed by the cell, and what we're left with at the end is just a cell, just a little package of keratin, a sacrificial package of keratin on the surface of our skin. Now, it's worth emphasizing the water barrier functions of the epidermis. I've mentioned it several times already, but it's really hard to downplay this. We need to retain water. Generally speaking, assuming you're not in a desert environment, we need about a half a gallon of fluid intake per day. And of that fluid that we intake every day, most of it's going to be drunk or drink, we drink a beverage. But we also get fluid directly from the food we eat. Think of, grapes, watermelon, citrus fruits. There's a lot of water in that food, those food products. We also are going to make water through cellular respiration. But we need to keep all of that water in our bodies. Now, this waterproofing that we have is going to primarily function on keeping water in, and it's not going to prevent water from being reabsorbed or absorbed by superficial layers of the epidermis. So if you spend too much time in the bathtub, that stratum corneum is going to absorb some water. Some of those superficial layers of the stratum corneum will absorb water. You can get the pruny fingers, or if you have thick skin on your feet, a lot of times that thick skin will swell up, and that's a really good time to rub it with a pumice stone because that water that's been absorbed by the skin softens it up a little bit. Deep to the epidermis, we have the dermis, and this is going to be the living component of the skin. It's worth mentioning that this is the part of the skin that you need to get a tattoo in when you're getting ink done. You need to deliver the ink to this part of the skin. If you only have the ink for your tattoo within the epidermis, eventually it will be shed with the keratinocytes that are continually flaking off. But if you can get the ink down into the dermis itself is not sloughing off cells constantly, and the ink will remain, for years to decades. Now, in terms of the thickness of the dermis, it can be super thin, 1/5 of a millimeter, or 2/10 of a millimeter, or it can tend to be a little bit thicker, and half a centimeter almost, or 4 millimeters. So depending on the part of the body, the dermis is highly variable in thickness. The single most common component by mass within our dermis is going to be the collagen protein. And those collagen fibers are oriented in many different directions because we have the dense irregular connective tissue, which makes up our dermis. We also are going to have lots of blood vessels. Being a connective tissue, or being made up of a connective tissue, the dermis is going to be very vascularized. It has a good supply of nutrients and a good ability to remove waste products. And because of its high nutrient supply, we're going to have most of the exciting things happen in the dermis. We're going to have our glandular secretions. We have most of our nerve endings. Most of our metabolically active parts of our skin will be housed within the dermis because this is the part of our skin that has the biggest nutrient supply. Now, as we're looking at the dermis, it's worth emphasizing we have hair follicles and nail roots housed within the dermis. The hair follicle and nail root are remarkably similar to each other in that they specialize in producing keratinocytes and filling those keratinocytes with keratin. The differences between them is the final shape assumed by those keratinocytes. One, the nail root, is going to take those keratinocytes and make the plate of our nail, our fingernails or our toenails. And the hair follicle is going to take those keratinocytes and extrude them as a cylindrical or tubular structure. But both are just made of a lot of keratin. We're also going to have, within the dermis, a lot of attachment points for skeletal muscle. Those collagen fibers of the dermis are going to be anchored to the underlying epimysium of the skeletal muscles so that we can wiggle our muscles and have facial expressions. Or for those of you who are really lucky, you can wiggle your scalp or wiggle your ears if you have really good fine motor control. Within our dermis, we have some wavy boundaries. And that's a superficial wavy boundary that goes up and down, up and down. Each little bump is referred to as a papillae. And papillae is a very generic term in the human body. We have lots of papillae. And we refer to these specific papillae as dermal papillae. And this region of the dermis that's made of the dermal papillae is known as the papillary region of our dermis. And what do these papillae do? What do these ridges do? They help to anchor the epidermis to the dermis. They also give us fingerprints. Very large, very pronounced dermal papillae on our hands and our feet give us our footprints, well, I should say, the prints of our feet. And they also give us our fingerprints, those friction ridges on our hands and our feet. So as we look at that papillary layer, it is the superficial layer of the dermis made of dermal papillae, and it's going to focus on allowing the dermis to be anchored. It's also going to have a lot of blood vessels. It's highly vascularized. A small cut to the papillary layer, a small amount of damage to the papillary layer, can cause a lot of bleeding. You've noticed if you ever had a tiny little scratch, that tiny little scratch can cause a lot of blood flow. I'm thinking of a paper cut I had earlier this week. It bled for about a half an hour, primarily because of all of those capillaries within the dermal papillae, or the papillary layer. We have deep to the papillary layer the reticular layer of the dermis. And this reticular layer is going to have a different orientation to the collagen fibers. Within the papillary layer, we have primarily areolar tissue. Within the reticular layer, we have that dense, irregular connective tissues. So the papillary layer within areolar tissue, you can think of as kind of a glue. And whenever you see areolar tissues in our body, I want you to think of an adhesive tissue. It helps to anchor two separate tissue types together. In particular, the areolar tissue of the papillary layer will anchor the stratified squamous epithelium of the epidermis to the dense, irregular connective tissue of the reticular layer. And as we're looking at this reticular layer, this is where stretch marks will form in our bodies. Generally speaking, those collagen fibers are going to be oriented in many different directions. But if you take the dermis and you stretch the dermis out, all the collagen fibers will be parallel with each other. And when those collagen fibers have a parallel orientation to each other within the dermis, it visibly manifests itself as a stretch mark that you can see in our body. Stretch marks will usually appear in the armpits, the butt, particularly for women who have lactated. They'll appear on the breasts, parts of the body that have dramatically increased in volume in a short period of time are prone to developing stretch marks. So as we're looking at these layers, within the papillary layer, made of that areolar tissue, we have smaller collagen fibers, and then within the dense, irregular connective tissue of the reticular layer, the collagen fibers are much more pronounced and emphasized, much stronger. Deep to the dermis, we have the hypodermis. The hypodermis, there's just no good place to put it. It's technically not part of the skin, but because we don't have an organ system really to group it with that's better than the integumentary system, it's traditionally grouped with the integumentary system. And as we look at our hypodermis, it's going to consist primarily of adipose tissue and areolar tissue. It's going to have this function of padding the body. When we think of that adipose tissue, I want you to think of cushioning. It's a shock absorber. And when we think of the areolar tissue, I want you to think of that glue that will help to bind things together. Another name for the hypodermis is the subcutaneous region. Sub meaning deep, cutane or cuticle referring to skin. So hypodermis and subcutaneous are synonyms with each other. And when somebody's having a subcutaneous injection, that's typically going to be drug delivery to the adipose tissue of the hypodermis. And they'll deliver drugs to those parts of the body when we want the medication to slowly diffuse throughout the body. Adipose tissue has a low blood supply. It has a little bit of vasculature, it has a little bit of blood vessels, but not a lot of blood vessels. And because it has that moderate amount of blood vessels, if we receive a vaccine or some kind of other injection into that subcutaneous fat, it's going to percolate and diffuse throughout the body slowly over a period of days, as opposed to an intravenous injection, which will diffuse throughout the body in a period of seconds because it's delivered directly to the bloodstream. Now, as we look at that subcutaneous fat, a big one is energy reserve. We use subcutaneous fat to help store calories long term, but it's also good for thermal insulation. You've probably spent time at a public swimming pool and looked at that 5-year-old kid that's super skinny. They maybe weigh 30 pounds soaking wet, and they're just shivering the whole time. Even though it's the middle of summer, they have blue lips. And one of the reasons for that is because they have almost no thermal insulation, or they have almost no subcutaneous fat. When we look at the subcutaneous fat, one of the sex differences is that traditionally in women, there's a thicker layer to the hypodermis. There's a little bit more adipose tissue within the hypodermis of females compared to males. A primary reason for that is the females, generally speaking, whether you want to or not, physiologically, you're going to be prepping your body for pregnancy. And pregnancy is something that requires a lot of reserve energy. And if there's not enough reserve fat within the hypodermis, the menstrual cycle shuts down because you don't have enough reserve energy for pregnancy. When we look at infants, generally speaking, when we say infants, we're referring to little kids like 2 or 3-year-olds, brand new born babies that are wet behind the ears. They're pretty chunky and they're super cute. But once you hit the 3 to 6-year-old age range, if you lost the brown adipose or the brown fat, generally speaking, there's not that much adipose tissue within young children. Another population that loses that subcutaneous fat is the geriatric population. It's very common for people a few years before they pass away to lose most of their subcutaneous fat. In particular, a lot of people have rapid weight loss right before they pass on. So let's pause for a moment and have a review question. I'm going to give you a couple extra seconds to this one. So I'll give you an extra 60 seconds. This is a sorting question. Please sort the layers of skin from superficial to deep. So you have 2 total minutes on this one. Please talk to each other. Let's chat. Let's make sure we can get some communication going. So we're down to about 1 minute. Down to about 1 minute. We need to orient these layers from superficial to deep. So we're down to 15 seconds. Make sure you submit your answers so it gets recorded. There we go. All right. So let's talk about the layers. Starting from superficial to deep, we have the stratum corneum of the epidermis. Deep to the stratum corneum, we have stratum lucidium. After stratum lucidium, you should have stratum granulosum. From there is to be stratum spinosum, and the next stratum basale. So those are the 5 layers of the epidermis. And now we're going to move on to the 2 layers of the dermis. The most superficial layer of the dermis is the reticular layer, the papillary layer. And then deep to that is the reticular layer of the dermis. So in terms of what it should look like, this is the correct order for that question, from superficial to deep. So we've talked about the epidermis and those 5 layers on the surface. We've talked about the dermis. We've talked about the hypodermis. Let's focus on some of the specialized structures within. First, let's focus on skin color. Melanin is the pigment that is produced by the melanocytes and gives us most of the coloration of our skin. You should make a side note. Melanin is not melatonin. Melatonin is a hormone that makes you sleepy. Melanin is a pigment which gives you a nice tan or deep dark coloration to your skin. And, I understand they sound super similar to each other, but they're very different things. So melanin is a pigment and melatonin is a hormone. So make sure you make a margin note of that so you don't get them mixed up. Because every semester, there's usually about 10% of the students will say melatonin when they meant to say melanin. So as we look at melanin, this pigment that gives us coloration to our skin, there's 2 forms of melanin. We have eumelanin and phenomelanin. When we look at eumelanin, the prefix EU means true. So eumelanin or true melanin is the most common variation of melanin, the first discovered and the most noticeable pigment or form of melanin. And this eumelanin gives dark coloration to the skin. So if somebody has, brown or black skin, they're going to have high concentrations of eumelanin being produced by the melanocytes of their skin. Another variation of melanin that's made in much smaller quantities is called phenomelanin. And this one was not discovered until relatively recently, relative to the eumelanin. This phenomelanin is going to have a reddish coloration to it. Sometimes it's described as kind of a yellowish coloration, but most people will define it as more of a reddish coloration and leave the yellowish portion or pigmentation you can see in the skin to the keratin within the keratinocytes. So I think this is kind of interesting, which is why I gave it special emphasis. Regardless of your skin color, you can be the whitest white person or the blackest black person or anywhere in between, we all have the same number of pigment-producing cells. The difference between fair skin and dark skin isn't the number of melanocytes, it's how active our melanocytes are. Individuals that have naturally darker pigmentation naturally are going to have more of a hormone known as melanocyte-stimulating hormone circulating in their bloodstreams. And the more melanocyte-stimulating hormone you have, the more melanin the melanocytes make. So if you want, and this has actually been a thing, it's really expensive, but if you want to get the best tan of your life, without the fake baking, you could go and get an injection of MSH, melanocyte stimulating hormone, and it won't be, be the perfect tan, and it'll be an instantaneous perfect tan over the course of a couple days as your melanocytes automatically start making more melanin. For most people, though, it's not very cost effective. I recommend just spending more time outside, personally. So really, when we look at skin colors and skin pigmentation, it's not the number of melanocytes, it's the concentration of melanocyte-stimulating hormone within the bloodstream. And as we're looking at the difference between someone with dark skin and someone with fair skin, all of those color differences within the skin are going to be primarily concentrated within the stratum basale of the epidermis. So within lab, we had the slides labeled person of color. The technical term for that is negroid, and when we had the white skin person, the technical term for that is caucasoid. And as we're looking at those layers, those skin pigmentations, the melanocytes are going to deposit melanin within the keratinocytes of the stratum basale. And that's where most of the pigmentation is going to be concentrated to give color to the skin. Something else that's going to influence skin color is exposure to ultraviolet radiation. As we have more and more UV exposure, our melanocytes are going to upregulate melanin production in an attempt to minimize ultraviolet damage to our genome. And then when we spend, the last 3 months indoors and avoiding the sun, because there is no sun, that melanin that's produced will be sloughed off as we lose keratinocytes, and our skin will begin to pale. It typically takes about 30 to 40 days for us to lose a tan, because that's how long it takes for us to shed the keratinocytes from our from our epidermis. Now, there's more than just melanin, the eumelanin and the phenomelanin. There's hemoglobin and keratin. Hemoglobin is the red pigment within our blood. People that have a ruddy complexion have a lot of superficial blood vessels that give them a reddish pigmentation to their skin. And then individuals that have lots of keratin in their skin are going to have more of an orangish yellow coloration to their skin. One of my favorite fad diets was the pumpkin diet. And people that followed the pumpkin diet ate lots of orange pumpkin, orange carrots, orange squash, and the really good people, the really motivated people, you could tell they were really motivated because their skin had excessive amounts of keratin concentrating in it, and they started to turn orange like a pumpkin. Needless to say, that fad diet died out pretty quickly. Most people didn't want to look orange, even if they lost some weight. And let's face it, if you were only eating carrots and pumpkins, you'd lose a lot of weight, because I would never want to eat. But there's more than just those pigments to our skin. Skin color is important from a diagnostic perspective. So we have a bunch of aspiring healthcare providers. Blue skin is technically referred to as cyanosis. And this is a blueness due to oxygen deficiency. A lot of times, superficial tissues of the body will have oxygen deficiency as we vasoconstrict the capillaries near the surface. So when somebody is really cold, their lips will appear blue because they don't send oxygenated blood to the lips. They're sending that warm blood to the core of the body to keep the core organs at operating temperature. We also could have erythema. And this is at the root word ureth for erythrocyte or red blood cell. If you have lots of erythrocytes or red blood cells near the surface of your skin, you look really red. Erythema is usually associated with overheating or strong emotions. We also could have pallor, and I wanted initially to go with white font for pallor, but white on white is hard to read. When you have pallor, that's going to be an absence of blood near the skin. The skin is going to be very white in appearance due to dramatically decreased blood flow within that tissue. And pallor is differentiated from albinism because pallor is whiteness due to lack of blood flow. Albinism is very white skin due to the lack of melanin. If that person has an inability to produce melanin, they have a mutation that makes their melanocytes unable to make that pigment, their skin is going to be very white. Their eyes tend to be blue, gray, occasionally red, depending on the severity of their albinism. And they tend to have white or almost yellowish hair because they'll still have a very high keratin content to their hair. So usually near the roots, it kind of looks yellow, and then, a couple inches away from the root, it'll start to appear white due to the lack of pigmentation. Every once in a while, you'll see somebody that's albino, and they don't look white. They're cherry red. And that's because they sunburn on the drop of a dime. I remember when I was an undergrad, I spent some time in the Bahamas studying coral reef ecology. And I was in the Bahamas, and I met an albino individual. And there's too much sun in the Bahamas. I remember I got sunburned through my denim blue jeans while I was down there. And then I met this albino individual, and at first I didn't know he was albino, and then I started chatting with him, and I noticed the white hair, the red eyes, and his skin was just cherry red. And I remember thinking to myself, I would never live this close to the equator if I was an albino. I would move up to Alaska. But, to each their own. If somebody has yellow skin, that's referred to as jaundice. That's due to an excessive amount of bilirubin that builds up in the blood. The best way to get rid of bilirubin is to defecate, to go number 2. Usually newborn infants will have jaundice. It's not uncommon. And when they, that first time they defecate, the meconium, or that thick tarry poop they excrete, is highly concentrated in urochrome, which is a byproduct of bilirubin processing. We also could have purple or blue or yellow. I really should go almost like a rainbow font for the hematoma, because a hematoma is a bruise, and bruises will be purple, red, yellow, green. There'll be all kinds of colors as those subdural hematomas are processed. And then finally we have bronzing. And I would describe bronzing, and I'll get on a bit of a high horse on this one. I know we're kind of pressed for time, but this is a personal one for me. Bronzing is when somebody becomes abnormally tan. And I'm going to go as far to say not just abnormally tan. They become freakishly tan when there's no good reason for them to be freakishly tan. They become unbelievably tan even though they never go outside into the sun. A key symptom of bronzing and the associated Addison's disease is that their gum line becomes tan. And if you're a Caucasian and your gums turn black because you're producing so much melanin, that's a giant red flag, because Caucasians should never have black gum lines from excessive melanin production. Addison's disease is one of those endocrine or hormonal disorders that's exceedingly rare. Most doctors learn about it a little bit in med school, and then they never see it again and forget about it. I just happened to know a young person who had it, and because of the Addison's disease, he spent a summer passing out spontaneously due to salt imbalances in his bloodstream, and he went through 4 doctors before he was correctly diagnosed with the disease. So just as a heads up, if you're ever, in the future working with a Caucasian who lost lots of weight rapidly without trying, and they have a tan gum line, that's a giant red flag. They have the hormonal disease of Addison's. It's very, very rare, though. You'll probably never see it. Hopefully never see it. So let's talk ancestry of skin color. Our skin color, our default pigmentation level in our skin, is determined by our genetics. Our genetics are determined by our parents, and their genetics were determined by their parents, so on and so forth, back through the generations. And generally speaking, depending on where our ancestors are from, that's a good indication of what our skin color is probably going to be. Not guaranteed, but probably going to be. So if we have ancestors, or most of our ancestry is going to be from individuals that are near the equator, we tend to have darker pigmentation because individuals near the equator are traditionally going to have more ultraviolet exposure and consequently will need more melanin production to downregulate damage to their superficial nuclei of their cells to minimize the mutations. Individuals that are kind of around the 45th latitude north or south, typically north because there's not that much land mass on the south 45th latitude, So, individuals near Europe, the Midwest, if you have a lot of European ancestry, those individuals didn't have to worry about excessive sun exposure. Instead, their problem was not enough sun exposure. So consequently, in order to maintain enough vitamin D in their bodies, preferentially, they had natural selection for individuals that had less melanin production. So we typically have pale skin individuals that come from those parts of the world or that ancestry. And you're probably wondering yourself, well, what about Eskimos or people that are from those really high northerly latitudes traditionally? How come they traditionally have darker skin? Because those high northerly latitudes don't necessarily have a lot of direct sun exposure. But the catch-22 is there's lots of snow. So that even though they may not have a lot of direct sun exposure, individuals whose ancestors are from traditionally snowy regions will have light reflecting up off the ground, off the snow, and hitting them in the face. So they actually have amplified UV exposure. If you've ever spent a day skiing on the slopes, you may have noticed how a lot of times people can get sunburned in the middle of winter if they spend all day skiing and their cheeks are exposed. So I jumped ahead. I stole my thunder here. So populations from far northern and southern latitudes need to have lighter skin for adequate UV. Unless you're super far north and have that sunlight reflecting off the snow, then your ancestors traditionally would have a little bit darker pigmentation, usually more of a brownish pigmentation. Why do we need vitamin D? What's the big deal with vitamin D? Well, vitamin D primarily is going to be associated with calcium processing. Not only does it allow us to absorb calcium from our food and our digestive tract, it also allows us to reabsorb calcium from our urine or to filtrate as we're making urine. If we don't have enough vitamin D, we don't have enough calcium, and then those things that we need calcium for, we struggle with. In particular, females will, generally speaking, lactate after giving birth. We're mammals. We have breasts, and we give our infants milk as part of, our mammalian heritage. And women, or females that are going to be lactating or pregnant, need a lot of calcium in order for the skeleton of the child in the womb to be developed, and then they need a lot of calcium in order to put that calcium in the breast milk for the child as the child has rapidly ossifying bones. Some other things that are going to influence skin color include just cultural differences with clothing. If traditionally your ancestors wore a loincloth, they had a lot of sun exposure, and they would have had selection for more pigmentation. And then on the flip side, if traditionally your ancestral heritage, or your traditional ancestral garb was to be completely covered up, your ancestors would have had less sun exposure, and that would have favored or selected for the fairer skin. Something else that influences skin color is intermarriage. Increasingly, being able to say, I'm of African descent or European descent or of South Pacific descent, and only one area is becoming increasingly rare as we have more international travel and more marriage across different ancestry lines. So we're having multiracial children. So we're having a blending of the skin colors. And then finally, there's sexual selection. Depending on your own personal preference, some people prefer to have a partner with darker skin. Some people would prefer to have a partner with lighter skin. And some people don't care at all. But there is a sexual selection component as well. Let's talk about markings on our skin. Those friction ridges or fingerprints on our skin are going to be determined while we're in utero. And I think this is kind of cool. Even identical twins with identical genomes have different fingerprints. Because it's not only your genetics, it's also the environment of your development. This is a nature-nurture thing. Genetics play a component in your fingerprint determination, but the flow of amniotic fluid in utero also determines the development of your fingerprints. And when we have identical twins in the womb, they have one's in front, one's in back, one's on top, one's on bottom. So there's going to be different pressures, different flow rates of the amniotic fluid, and that's going to influence the development of friction ridges as the dermis is developing in utero. What do we use friction ridges for? Well, to grab things, or the manipulation of small objects. They increase friction. Fingerprints make its way of more friction, so you can grab smooth and slippery things and not have them fall out of your hand. They're a good thing. We also have flexion lines, and these flexion lines are just natural creases in the body. So if you ever look at your palm, you can see that there's some deeply embedded lines in the palm of your hand. Those deeply embedded lines are flexion lines, and these flexion lines are preferred directions that our skin prefers to bend. And in terms of a clinical significance, when you are ever going to be working with a patient and you're making an incision on the patient, you always want that incision to be parallel with their flexion lines, because it's easier to stitch up and there'll be a higher success rate of healing the injury or the incision. If an incision is ever made perpendicular to a patient's flexion lines, that incision has a tendency to rip open constantly and will heal slower. So learning the flexion, if you're ever going to be doing any kind of cutting on a human being, you need to learn the flexion line so you can cut in the right direction. We have freckles and moles. We all know what a freckle and mole is, but let's get technical with what a freckle or mole is. A freckle or a mole is caused by an abnormal accumulation of melanocytes. So that dark dot will be from a bunch of melanocytes that are producing a bunch of pigmentation, either melanin or eumelanin. So how do we differentiate freckles versus moles? Freckles are two-dimensional, moles are three-dimensional. So freckles are flat, moles stick out. The technical name for a mole is a nevi. I've never actually heard anyone ever use the term nevi in conversation, but I just wanted to throw it out there. That's the technical term for a mole. When we look at moles in particular, you need to watch the color, diameter, the hairs of the mole. Moles have a tendency of becoming cancerous. So you need to monitor moles to make sure they aren't rapidly changing. Generally speaking, over the course of our lifespan, if you have fair skin, if you're naturally a Caucasoid, you are going to accumulate more DNA damage to your skin over the course of your lifetime. And as you accumulate more DNA damage because you have less pigment to protect your skin, you're going to have more moles and more freckles develop over the course of your lifetime. So for me, speaking personally, before I was in high school, I had a completely bare back. But after spending 2 summers lifeguarding while I was in high school, now I have moles and freckles all over my back because I wasn't very good about using sunscreen while I was lifeguarding. Now, we also could have a birthmark or hemorrhagia. And sometimes these are referred to as strawberries. These birthmarks or hemorrhagias are going to be caused by lots of capillaries within the dermis growing out of control. It's an abnormal concentration of blood capillaries within the dermis. So they tend to be pink and three-dimensional. Generally speaking, birthmarks fade as we age. And we go from having a big glob of three-dimensional capillaries within our dermis. As we grow and increase in volume and size, those capillaries stretch out and go from being three-dimensional to two-dimensional, or they go from being visible to not visible. So these birthmarks tend to fade as we age, as we increase the surface area of our skin. We also have hair and nails. These are some accessory structures that have high concentrations of keratin. It's worth noting, and this is just connecting to the news recently, I read in the news 2 days ago that we are down to 2 white rhinos living in the wild in the world. So they're basically extinct at this point. It's unfortunately almost inevitable because we have only 1 male and 1 female left. So even if they did reproduce, there'd be inbreeding issues with the offspring. And that rhinoceros horn that they've been poached and killed for is just made of keratin. And people who like to consume rhinoceros horns are, from a biochemical perspective, eating expensive fingernails. And we've made the white rhinoceros practically extinct, well, functionally extinct at this point, because some people like to eat the keratin of the horn. So as we look at the hair and nails, our hair, fingernails, and toenails, These are accessory organs or appendages with high concentrations of that keratin. Now, there are different kinds of keratin. We can have hard keratin and flexible keratin. We're primarily going to have a flexible or pliable version of keratin within our epidermis. And then within the hair and nails, we have a tougher version of keratin that has more cross-linking. And because there's more cross-linking, it's harder to pull that keratin apart, and it tends to be stiffer and more rigid. So as we're looking at a hair, the technical name for a hair is a pilus or a pilus. To me, it's a potato-potato situation. So if we have multiple hairs, it's a pili. Singular is pilus or pilus. That's the technical name for a hair. And as we're looking at this hair, it's a filament of moderately flexible keratinized cells. So it's kind of an in-betweenie. Our skin has only flexible keratinized cells. Our nails have very rigid keratinized cells. And then there's our hair, which is a combination of both. It's both rigid and flexible at the same time. So where in our bodies do we not have hair? The general rule of thumb is genitals and thick skin. If you really want to just get it down to its bare basics. So with the male anatomy, the penis, the shaft of the penis, the head of the penis, known as the glans penis, that's going to be free of hair. And then palms of the hands, bottom of the feet. And then for the female anatomy, it's going to be the labia minora and the shaft of the vagina, what's also referred to as the vestibule, or that opening in between the labia minora, that's also free of hair. Other parts of the human body that don't have hair include nipples and lips. Just parts of the body where having hairs would hinder the function. Remember, structure determines function. And having hairs in those parts of our body would be disadvantaged or not advantageous. When we look at our arms and trunk, we have a moderate concentration of hairs. And regardless of your sex, you male or female, we have the same concentration of hairs. The difference between somebody who looks like a gorilla and somebody who has almost no body hair at all, or no visible body hair at all, isn't going to be the concentration of hairs. It's going to be the activation of those hair follicles. So I remember a guy I lived with. I rented a garage from him when I was in college. Great rate of rent when you live in a garage. And I remember once I saw him in the summer, and I thought he was wearing a sweater in the middle of summer. But it just turns out he's naturally very hairy. He's one of those guys that, just is oozing testosterone. So he's completely bald on top. And then all the hair on top seems to have migrated to the rest of his body. And then you could look at individuals that have low testosterone concentrations. Having a low testosterone concentration means that those superficial hairs aren't going to be thick and black, but instead will be fine and white and hardly visible at all. A good rule of thumb is that if you can get a pimple in that part of your body, you have a hair follicle in that part of your body. Because all pimples, all acne, are from hair follicles that have a bacterial infection. So when we look at our arms, legs, and trunk, we have a relatively low concentration of hair. But then our scalp has a much higher density of hairs. So we're looking at about 100,000 hairs per an average scalp, or to put it in terms of hairs per square centimeter, we're looking at about 100 hairs per square centimeter on your scalp. So as we're looking at a standard generic hair, there's going to be the bulb. The bulb is going to be deep within the dermis. It's a bulging, hence its name, bulb. And within that bulb, we have the living hair cells. This is the part where the hair is actively reproducing. So when somebody pulls a hair out of your body, maybe you're getting waxed, maybe you're getting tweezed, maybe you have little kids that just pull your hair constantly. Whatever the reason, when that hair gets pulled out, the bulb gets damaged. The living part of the hair gets damaged. And within that bulb, we have a nerve ending that gets torn. That's why it's painful to pull out hairs. However, we've all had a haircut. It typically doesn't hurt to shave. It doesn't hurt to get a buzz or a trim because those parts of the hair are non-living and don't have metabolically active sensory receptors in them. Now, as we're looking at the root of the hair, this is typically going to be defined as the part beneath the surface. The shaft is typically defined as the part above the surface. We're also going to have wrapped around the bulb, wrapped in the middle of the bulb, something known as the dermal papilla. So this is that term papilla again, but this is a small little bud of the dermis within the center of the bulb that contains a high concentration of blood vessels supplying the nutrients for the hair. And then when we look at the body of the hair, so the part of the hair that makes up its reproducing section and the thickness, the volume of the hair, that's referred to as the matrix of the hair. So to look at our diagram in histology, we have the bulb on the bottom. We have, let me switch colors here. In green, we have that dermal papilla within the bulb, and then we have the matrix directly above the dermal papilla, which is where we have the actively reproducing cells of the hair that make up the vast majority of the hair's volume. If you were to take a hair and cut it in half, transversely or in cross-section, or even longitudinally, the three layers you would see are from deep to superficial are the medulla to the cortex and then to the cuticle. The medulla is the central shaft of the hair follicle, or the hair. A lot of times, the medulla will either be filled in or will be hollow, depending on your hair color. The cortex is going to be what is primarily made by the matrix, and the cortex is the thick volume of our hair. This is the part of our hair that determines if we have straight hair, wavy hair, or curly hair. It's all within the cortex. For those of you who have more of a biochemistry background, there's a special kind of bond called a disulfide bridge within the proteins of the cortex between two cysteine amino acids. And it's that disulfide bridge that helps to give the cortex its structural arrangement. And there's some neat chemistry here behind a perm. When you're getting a perm, you take highly alkaline chemicals, put it on the hair, and those highly alkaline chemicals will temporarily break the disulfide bridge. Then you can take the hair, wrap it around a curler, and then add some vinegar or citric acid to reacidify the hair. You reform the disulfide bridges, and now you've rearranged the hair from straight to curly. Or you could take the curly hair and make it go from curly to straight, depending on its orientation when you reacidify the hair after you add that highly alkaline compound to it. It's also the same chemistry behind Drano, because that strong base will tend to dissolve and make hairs that clog your drain more pliable so they can get flushed down the drain. Around the outside of the hair, we have the cuticle. And as we look at the cuticle of the hair, we have some very thin, scaly cells that have some free edges that act like friction ridges or act like mild barbs. And these free edges are directed upward as the hair grows out, but then as the hair flops over, it's almost like shingles on a house or shingles on the roof of your house, because the free edges are going to overlap and tend to point downward and help direct water down off of your hair as opposed to directing the water toward your scalp. So let me jump ahead here a couple slides. So to back up here, we have the medulla in the middle. We have the cortex that makes up the main body. And then around the perimeter, we have the cuticle of the hair to look at those three layers in cross-section. What the hair follicle itself is going to be the part of the hair that extends down to the dermis, occasionally into the hypodermis. And this tube is going to help to support the growing hair root. It's going to have an epithelial layer to its sheath. It will have some stratified squamous cells that make up its sheath and will lie around the hair. This stratified squamous epithelium of the hair follicle is also a reserve source of multipotent stem cells. if you ever look at a burn victim or somebody who's lost their skin because of abrasion or burn, you'll notice that as the skin is regenerating, new stratified squamous epithelium will grow up out of the hair follicles, and then during this process, it's known as granulation, they'll have speckles of skin as their skin is regrowing. And each hair follicle serves as a site of origin for new epidermis to regenerate from because of the epithelial cells within the hair follicle. We're also going to have some connective tissues that help to bind that hair follicle to the surrounding dermis, that denser regular tissue of the dermis, to help hold it in place. We have one nerve ending per hair follicle, so when you pluck a hair, it hurts, especially the first time. But if you're a veteran, if you've been plucking hairs for years or for a long period of time, eventually those nerve endings stop regrowing, and eventually it gets to the point where it doesn't hurt to pluck those hairs anymore. But the first time is always very painful. We also have an erector pili muscle that is going to reorient the hair follicle. That erector pili muscle, sometimes referred to as a piloerector muscle, is going to give us goosebumps. And when that smooth muscle tissue is activated, the hair follicle will go from kind of a diagonal orientation to a perpendicular orientation and jut straight upward from our skin. Now, if you're an individual that doesn't have a lot of testosterone in your body, in your bloodstream, you're not going to have big, thick black hairs all over your body, and goosebumps don't serve a really good purpose. They're kind of a vestigial function. But if you're a very hairy individual, goosebumps will make it so that all those hairs all over your body help to trap air and help you to get just a little bit warmer. Not that much warmer, because we aren't really that hairy anymore as a species. And it's time. I'm going to call it. My watch says 15 after.